Welcome to Genomic Medicine, your shortcut to being well-informed in the world of cutting-edge biology. Today, we're embarking on a deep dive into, well, one of the most exciting and rapidly evolving fields in modern science. Have you ever wondered why two people, maybe even identical twins with almost identical genes, can have such wildly different health outcomes, even when facing, similar challenges? Or, think about this for a second. What if the lifestyle choices your grandparents made decades ago could literally affect your own predisposition to certain diseases today? It honestly sounds a bit like science fiction, doesn't it? But today we're going to unpack an incredible dynamic layer of our biology that holds some, well, truly surprising answers to these exact questions.
Yeah, it's fascinating because for a long time, our understanding of biology often felt very fixed, almost deterministic, you could say. We typically thought of our genes as this unchangeable blueprint, passed down through generations, fixed. But increasingly, the scientific community is recognizing there's a far more dynamic, adaptable dimension at play, something we call the epigenome. And this deep dive will explore how the second dimension of our biology profoundly impacts our health. It influences everything, really, from the earliest stages of our development to the onset and progression of complex diseases. And yes, even hinting at a revolutionary new understanding of inheritance across generations.
Right. So our mission for you today is to unpack this intricate world of epigenetics. We'll explore its fundamental role in establishing and maintaining the very identity of your cells all throughout your body, making sure, for instance, that your kidney cells behave like kidney cells and not, say, brain cells. That's crucial. Then we'll explore its surprising and critical connections to common and devastating diseases. We're going to focus particularly on cancer and neurological disorders today. And maybe most excitingly, we'll delve into the revolutionary potential it holds for future therapies. So you'll leave here with a clearer understanding, hopefully, of how your body's unique genetic expression isn't just about the DNA sequence itself, it's also about these constantly adjusting instructions that tell those genes exactly what to do, where to do it, and critically, when to do it. Okay, let's unpack this key concept then, the epigenome. We hear so much about the genome, that's our entire DNA blueprint, right? The complete set of our genetic instructions. But what exactly is the epigenome and how does it differ from that, fixed genetic blueprint we're all pretty familiar with?
Okay, so imagine your genome as an incredibly vast, comprehensive library, a huge library. This library contains roughly 20,000 protein coding genes. Think of these as all the books of instructions needed to build and operate a human body. And crucially, every single cell in your body, whether it's a skin cell, a heart cell, a neuron, it contains an identical copy of this entire library. It's the same set of books everywhere in your body. Now, your epigenome, it isn't the books themselves. It's not the DNA sequence. Instead, think of it like the skilled librarian and maybe the entire operational system for that library. It's what dictates which genes are open for reading and are actively being used at any given moment, and which books are tightly shelved away, silenced, inaccessible. So while you possess only one genome, which is, constant across all your cells, you actually possess at least 400 different epigenomes.
About 400. It's a lot more variation than I thought.
Exactly. There's a unique epigenome for each of your body's specific tissues and cell types. For example, your epigenome is precisely what prevents, say, a kidney cell from trying to perform the specialized functions of a skin cell, or a liver cell trying to be a bone cell. It's the underlying fundamental mechanism that guides the differentiation of embryonic stem cells, as they develop into all the specialized cells of your body. And it's also responsible for the daily replacement of cells in your adult body, like those constantly renewing cells in your bone marrow or your skin. Honestly, without the epigenome, we'd just be a chaotic mass of identical cells, unable to form complex tissues and organs. It's that fundamental.
So it's definitely not a static blueprint then. It's much more like a dynamic, adaptable set of instructions, one that changes based on a cell's identity and its environment, too.
Exactly. That's the key. It's dynamic.
Okay, that's a fundamental shift in thinking, isn't it? Now, here's where it gets really interesting for me. How does this software, this librarian, actually work at the molecular level? What are the actual mechanisms that are used to open and close those genetic books?
Right, the nuts and bolts. Well, this dynamic landscape, it's orchestrated primarily through a series of molecular changes in what we call chromatin structure. Chromatin is essentially how your DNA is packaged inside the nucleus of your cells. You have these long strands of DNA, right? They're intricately wound around these tiny spools, like thread on spools. These spools are special proteins, and the way the DNA is wound around them, it can be tightened or loosened. When it's tightened up, the genes are generally inaccessible, switched off. When it's loosened, they're open, ready to be read, switched on. And there are two main molecular players that achieve this precise control. First, we have something called DNA methylation. This involves adding a small chemical tag, specifically, it's a methyl group, directly onto certain building blocks of your DNA. These are called cytosine bases. This addition often occurs in specific regions known as CPG sites. You can literally think of these metal groups as tiny reversible sticky notes placed directly onto the DNA strand itself. And when these notes are present, particularly on a gene's control region, they can effectively turn that gene off, signal it to be silenced or repressed. Our cells have a pretty sophisticated machinery for this. There are dedicated writer enzymes that add these tags, DNMTs are called. Then there are eraser enzymes, TETs, responsible for removing them. And finally, there are reader proteins that recognize these methylation patterns and in turn influence whether a gene becomes active or stays silent.
So writers, erasers, and readers, like a whole communication system using these methyl tags.
Precisely. And what's crucially important about DNA methylation is that these patterns, they differ significantly between individuals. While we all share essentially the same basic genome sequence, we're all human, right? Our epigenomes are far more varied, much more personalized. In fact, individuals can vary far, far more at the epigenome level than they do at the genome level. And furthermore, these methylation patterns aren't fixed throughout life. They're dynamic. They can change and vary greatly over time in response to various cellular perturbations, various signals. This includes influences from your diet, encounters with microbes like the bacteria in your gut, and even cellular stress, whether that's psychological stress or, exposure to environmental toxins. This fascinating variability suggests that many of the phenotypic differences we observe between people, why we look and function differently, including our predisposition to certain diseases, might actually be based more on these adaptable epigenomic variations than just on differences in our underlying static genomic sequences. It's a truly personalized layer of biology.
Okay, that's methylation. What's the second major player you mentioned?
Right. The second major player is histone modifications. As I mentioned, your DNA is intricately wrapped around these specialized proteins called histones, like beads on a string. These structures are called nucleosomes. Now, the histones themselves, these protein schools, can be chemically modified in various ways. They can have different chemical groups like acetyl groups or methyl groups added or removed from their tails. These modifications act like a complex kind of histone code that directly affects how tightly the DNA is wrapped around those histones. Think of it like adjusting a dimmer switch, maybe, that controls the accessibility of the DNA. When DNA is tightly wrapped, it's generally inaccessible to the cellular machinery that reads genes, so effectively turned off. When it's loosely wrapped, the genes become accessible and can be expressed or turned on.
So different modifications send different signals, like open here or keep closed.
Exactly. For instance, certain histone marks, like one called H3K4M3, are typically found at the promoter regions of active genes. They act as a clear signal indicating that the gene is open for business, ready for transcription. Conversely, other marks, like H3K27NE3, are strongly associated with gene repression. They act like a closed sign, indicating a gene should remain silent. Sometimes, interestingly, both activating and repressing marks can even coexist in specific regions. These are called bivalent domains. These domains are particularly interesting because they seem to provide a potential for really quick activation or quick repression, which is crucial for the precise and rapid differentiation of stem cells as they decide, what kind of specialized cell they're going to become. And just like DNA methylation, these histone modifications are not static, they're dynamic. They're introduced by their own set of writer enzymes, H8Ts and KMTs, for example. They're removed by eraser enzymes, HDACs and KDMs. And then they're interpreted by specific reader proteins, like those containing bromodomains, which recognize acetylated histones. This intricate interplay writers, erasers, readers, it's absolutely essential for finely controlling gene expression in every single cell of your body, adapting constantly to its needs and environment.
Wow. Okay, this whole system sounds incredibly complex. All these layers, chemical tags, proteins interacting, but also, incredibly intelligent and adaptive. It makes me wonder, does it, in a sense, remember things? Like, can a cell remember its past experiences or maybe its lineage through this?
Absolutely. This is precisely the concept of epigenetic memory, and frankly, it's one of the most profound aspects of epigenetics. Our epigenome has this remarkable ability to memorize lifestyle events, environmental exposures, in basically every tissue and cell type throughout the body. And it's not just a theoretical concept. We see this in action all the time. For example, our immune system. It can remember past encounters with microbes, which influences how it responds to future infections.
Like vaccines.
Exactly, yeah. Vaccines work, in part, by inducing an epigenetic memory in our immune cells, training them. Similarly, metabolic organs like our skeletal muscle, our fat tissue, our liver, they can remember lifestyle choices related to diet and physical activity. And this impacts their long-term function and how they process energy down the line. And this isn't just short-term cellular memory we're talking about either. The stability of DNA itself is a factor here. It has a half-life comparable to long-term human memories. So while your genome sequence remains essentially unchanged throughout your life, your neuronal epigenome, through these stable histone modifications and the intricate gene regulatory networks they establish, can potentially store information long-term. This suggests that the molecular basis for our long-term memories, the ones that last for decades, maybe memories from early childhood, it doesn't involve changing the DNA sequence itself, but rather it involves changes in how those genes are expressed, orchestrated via these epigenetic modifications.
So memories might literally be written in these histone marks.
That's a leading hypothesis, yes, for long-term memory consolidation. Short-term, day-to-day cellular responses, on the other hand, seem more tied to the more transient changes, like fluctuations in hisnoacetylation levels. But what's truly mind-bending here is that this memory function might even extend to what's called transgenerational epigenetic inheritance.
You mentioned that earlier, the idea that my grandparents' lives could affect me.
Exactly. It suggests that significant lifestyle choices and environmental exposures of your parents, and maybe even your grandparents, might in some cases directly affect your own epigenome, and consequently your health predispositions. Imagine, for instance, if your grandmother experienced a severe famine. That extreme stress could potentially leave an epigenetic mark that influences your metabolic health even generations later. It's a powerful, slightly controversial, but incredibly fascinating idea. It fundamentally extends our understanding of inheritance beyond just the DNA sequence we inherit.
Wow, okay, so this remarkable system governs our cells, lets them differentiate, maintains memory. It's clearly vital for health. But it also raises, well, an unsettling question. What happens when this finely tuned system goes awry? What happens when the epigenome gets reprogrammed incorrectly, particularly in the context of disease where these instructions get corrupted?
That's the crucial flip side, isn't it? If epigenetics is so fundamental to healthy cell function, what happens when those software instructions become corrupted or misprogrammed? As our source material clearly states, most non-communicable human diseases have a genetic inherited as well as an epigenetic component. So while genetic predispositions are absolutely vital, these epigenetic alterations also play a significant, often independent role in disease development. Let's delve into two major areas where this is profoundly evident: cancer and neurological disorders. These are detailed extensively in Chapter 13 of the material we're looking at. So first, let's explore epigenetic reprogramming in cancer.
Right. For so long, we've primarily understood cancer as, well, a disease of genetic mutations, errors or damage in the DNA sequence itself that drive uncontrolled cell growth. How does epigenetics, this second dimension, fit into that well-established picture? Is it just a side effect or something more fundamental driving the disease?
That's an excellent point. And the traditional view of cancer as primarily a disease of our genome caused by the accumulation of DNA mutations, things like point mutations, translocations, deletions, amplifications. That's still absolutely fundamental. Genetic mutations are critical drivers, no question. However, this deep dive emphasizes that tumorigenesis also comes along with abnormalities in cellular identity, a changed immune response, different responsiveness to internal and external stimuli, and major changes in the transcriptome, all of which are based on changes of our epigenome. And importantly, in fact, most types of cancer carry mutations both in the genome and epigenome. Yes, this is a critical distinction. It means changes in the epigenome aren't just side effects of the genetic mutations. They are often direct drivers themselves. They are essential contributors to the complex process of cancer development. They fundamentally alter how cells behave, how they respond to their environment, how they interact with the immune system. You could almost say they provide the fertile soil in which genetic mutations can take root and really flourish.
Can you give us some specific examples then? How do these epigenetic changes actually manifest and contribute to the hallmarks of cancer? What does this look like inside a tumor cell?
Certainly. We see several key patterns of epigenetic dysregulation that are frequently observed in many different types of cancers, and they often work hand in hand with genetic mutations to promote that uncontrolled growth. One of the most critical changes involves aberrant DNA methylation. We talked about methylation earlier. In healthy cells, specific regions called CTC islands, especially those located at the promoter regions, the on switches of tumor suppressor genes, These regions are typically unmethylated. They're kept clear. This allows these genes, which act as protective brakes on cell growth and division, to function properly.
Right, their brakes need to be working.
Exactly. But in cancer, we often see these CPG islands become hypermethylated. Too many methyl tags get added, like too many sticky notes piling up, gumming up the works. This hypermethylation effectively silences these vital protective genes. A really powerful example of this is the hypermethylation of a gene called MLH1. When MLH1 gets silenced epigenetically, it impairs the cell's DNA mismatch repair process. This is the system that normally fixes errors made during DNA replication.
So errors start building up faster.
Precisely. This single epigenetic change can then lead to a massive increase in the accumulation of DNA mutations throughout the entire genome. It kickstarts a cascade of genetic instability that fuels cancer progression. And conversely, other parts of the genome, like repetitive sequences, which are usually heavily methylated to keep them stable and prevent chromosomes from getting scrambled, these often become unmethylated in cancer cells. This loss of methylation causes genomic chaos, increasing the risks of chromosomes breaking, rearranging, causing further gene disruptions, like taking the safety locks off.
Okay, so methylation goes wrong in both directions. Too much in some places, too little in others. What about the histone modifications?
Yes, we also frequently observe histone modification dysregulation. In many cancers, there's a significant imbalance in the enzymes that control histone acetylation, that loosening or tightening of the DNA spools we discussed. We often see a loss or reduced activity of the writer enzymes, the HATs, that normally add acetyl groups and open up chromatin for gene expression. And simultaneously, there's often an overexpression of the eraser enzymes, the HDACs, that remove these acetyl groups. The net result is an overall reduction in histone acetylation. This creates a more closed, compacted chromatin state, which can promote the silencing of tumor suppressor genes and generally favor a cancer-permissive environment. And furthermore, it's not just the enzymes. Mutations in the histone proteins themselves, or their variants, can directly disrupt these critical patterns. There's a striking example. A specific mutation in a histone protein called H3.3. It's found in a really high percentage of certain bone cancers, chondroblastomas, and also some brain cancers in children.
Mutation in the spool itself.
Exactly. This single change in a histone protein can drastically alter the surrounding epigenetic landscape, leading to the misregulation of hundreds of genes and directly driving the cancer's development. It just shows how powerful these subtle epigenetic changes can be. Another common hallmark we see is the reactivation of pluripotency.
Pluripotency, like stem cells.
Yes, exactly. Cancer cells often undergo a process of dedifferentiation. They essentially revert to a more primitive stem cell-like state. This state is characterized by uncontrolled proliferation and a loss of their specialized functions. They forget what they're supposed to be doing. And this dedifferentiation is directly linked to the aberrant, inappropriate reactivation of genes that are normally active only during early embryonic development. Genes like SOX2, NANOG, OCT4, these are the genes that guide the formation of all cell types initially. Their reemergence in adult cells essentially puts the cell into an uncontrolled growth program. It's like hitting a reset button on cellular identity that promotes the rampant, unspecialized growth we see in cancer.
So they kind of go back to being like rapidly dividing embryonic cells, but in the wrong place and time.
In a way, yes. And the epigenome also plays a crucial role in genome instability and telomeres, contributing to the immortalization of cancer cells. You know how in normal cells, with each division, the telomeres, those protective caps at the ends of our chromosomes, like the plastic tips on shoelaces?
Right, they get shorter each time.
They do. And this shortening normally acts as a cellular clock. Eventually, it signals the cell to stop dividing or undergo programmed cell death, apoptosis. It prevents unlimited proliferation. However, cancer cells often manage to escape this crisis. They find ways around it. A key mechanism is the aberrant reactivation, often through epigenetic changes, of the gene that encodes telomerase. Telomerase is an enzyme that rebuilds and maintains telomere length.
So they keep rebuilding their chromosome ends.
Exactly. By reactivating telomerase, cancer cells gain virtually limitless replicative potential. They essentially become immortal in terms of division count. This process, along with other large-scale genomic rearrangements like chromothripsis, which is like the shattering and chaotic reassembly of parts of chromosomes, all this leads to rampant genome instability. And this instability is a hallmark shared not only by cancer, but also, interestingly, by the aging process itself, underscoring a deep connection between the two. And finally, epigenetic changes contribute significantly to immune evasion. how the tumor hides from the body's defenses, the tumor's microenvironment, that complex network of cells, blood vessels, signaling molecules surrounding the tumor, it shifts dramatically during cancer progression. In early tumors, you might still see immune cells that are trying to fight the tumor, doing their job. But in aggressive cancers, there's often a dominance of immune cells that actually support the tumor. They get co-opted.
Immune cells helping the tumor. How?
Well, cells like certain types of macrophages, called TAMs, along with immune-suppressive T regulatory cells, Tregs, and myeloid-derived suppressor cells, MDSCs, they become prevalent. These cells actively secrete various signaling molecules, cytokines and chemokines, that create a pro-inflammatory environment within the tumor. And critically, they also suppress the effective anti-tumor immune response. They actively shut down the killer T cells that should be attacking the cancer. This helps the tumor evade detection and elimination by the body's natural defenses. It's almost like they provide a cloak of invisibility or actively fight off the body's guards.
That's a truly comprehensive and frankly unsettling picture of how epigenetic errors contribute to cancer, intertwining with the genetic changes we already knew about. But what about the brain? You mentioned neurological conditions earlier. How does this intricate dance of epigenetic regulation influence things like Alzheimer's, Huntington's, or even developmental disorders? These conditions are often so devastating and currently have such limited treatments.
Right. The brain is another area where the epigenome is profoundly important. For normal brain function, for neuronal plasticity, that's the brain's ability to adapt, learn, form memories, and just overall neurological health. So when these epigenetic mechanisms are disrupted, it can have really severe consequences for the central nervous system. It can impact everything from early development to cognition later in life. One key area, similar to cancer in some ways, is histone acetylation imbalance. Many neurodegenerative diseases seem to involve disruptions in that delicate balance between the enzymes that add acetyl groups to histones, the HATs, opening chromatin, and those that remove them, the HDACs, closing chromatin. This imbalance results in abnormal histone acetylation levels in neurons. A prime example mentioned in the material is Rubinstein-Tybee syndrome. It's actually a monogenetic disorder caused by mutations in a gene called CREBP, which encodes one of those HET writer enzymes.
So they can't add the open marks properly.
Exactly. The mutation leads to abnormally low histone acetylation, and this contributes directly to the syndrome's characteristic cognitive impairment and physical challenges. We can even see this illustrated in figure 13.3 from the source. And similar imbalances in histone acetylation are implicated in a range of other neurodegenerative conditions too. Things like the polyglutamine diseases, such as Huntington's disease. It's also linked to the kind of transcriptional silencing, the inappropriate shutting down of genes that we observe in conditions like Friedreich's ataxia and Fragile X syndrome. Genes crucial for neuronal function get switched off, impacting the brain's ability to operate correctly.
So problems with acetylation seem quite common in brain disorders. What about methylation?
Yes, beyond these general histone modifications, specific methylation marks in neuronal function are also absolutely crucial for the brain's unique operations. For example, there's a specific protein, MECP2, that acts as a reader of DNA methylation marks. While it recognizes the conventional methylation at CpG sites, it also specifically recognizes methylation that occurs at non-CpG cytosines, so where C is followed by an A, T, or another C. This is often called MCA or MCA.
Non-CpG methylation, is that common?
It's surprisingly abundant in the brain, much more so than in other tissues. And genes that accumulate these non-CpG methylation marks during neurodevelopment, particularly in brain cells, they recruit this MECP2 reader protein. And the result is that these genes are subsequently repressed in their activity. This regulatory process seems vital for mediating distinct neuronal functions in adult brains versus newborn brains. It helps to fine-tune the mature brain's complex wiring and function. The importance of these non-CpG marks is further highlighted by an interesting observation. Between different individuals, these MCH marks are actually more highly conserved, meaning they're more similar across people than the more common MCG marks.
More similar. That suggests they're really important, right?
Exactly. This strong conservation underscores their probable profound importance in maintaining proper neuronal function and identity. It suggests they're critical for some fundamental brain processes shared across humans. Mutations in MECP2 itself, by the way, cause Rett syndrome, another severe neurological disorder.
Okay, and you mentioned memory earlier, too. How does that tie into neurological disease?
Right, and finally, we return to that profound role of the epigenome as memory storage. While we touched upon epigenetic memory generally, this is where it becomes particularly relevant for brain function. Our source material emphasizes that, quote, our neuronal epigenome remains as the exclusive device for information storage. Think about it again. Memories, especially those long-term ones that start forming around age 3 and can last for decades, they don't involve changes to your actual DNA sequence. Your genes don't change when you remember your first day of school. Instead, the persistent and stable changes in the neuronal epigenome, specifically through mechanisms like stable histone modifications, maybe methylation too, and the robust gene regulatory networks they establish, these are widely believed to be the molecular basis for long-term memory formation and maintenance.
So disruptions here could directly impact memory.
Precisely. Therefore, conditions that affect this epigenetic stability within neurons or disrupt the finely tuned balance of epigenetic marks required for memory consolidation and recall, these could directly impact cognitive function, learning, and memory. This could potentially lead to, or at least contribute significantly to, the devastating cognitive decline seen in many neurological disorders like Alzheimer's disease or other dementias, or even Parkinson's disease, which also has cognitive components. It gives us a whole new dynamic lens through which to view the molecular underpinnings of these complex brain conditions.
That's incredibly insightful, and it really is almost unsettling to think about how these subtle chemical adjustments, these epigenetic marks, can have such profound impacts on our health, our brain function, even our very ability to remember who we are. So if these epigenetic changes are happening and they're actively contributing to and even driving these complex diseases like cancer and neurological disorders, what does this all mean for treatment? Can we actually reverse these bad epigenetic programs? Can we sort of reprogram them for therapeutic benefit?
This is exactly where the field becomes incredibly exciting and holds, well, immense promise for the future of medicine. The fundamental principle here, highlighted again in the text, is that most epigenomic modifications are reversible, which implies significant therapeutic potential. Unlike genetic mutations, which are actual alterations to the DNA sequence itself, much harder to correct or reverse currently, epigenetic marks are inherently dynamic. They can be added or removed, tightened or loosened. That's their nature.
Because there are writer and eraser enzymes.
Exactly. This inherent reversibility opens up entirely new avenues for medical interventions and treatments. We can directly target the molecular hallmarks of epigenetic control, the writers, erasers, and readers we talked about, and we refer to these targeted treatments as epidrugs.
Epidrugs. Okay, what kind of epidrugs are we talking about here? How exactly do they work to reverse or modify these epigenetic patterns that seem to be driving disease?
Well, epidrugs primarily work by inhibiting those key enzymes, the writer enzymes that add the marks, the eraser enzymes that remove them, or sometimes the reader proteins that interpret these marks and influence gene activity. It's really about trying to reestablish the correct epigenetic balance that's been lost in the disease state. So let's look at HDAC inhibitors first. Histone deacetylase inhibitors. As we discussed in the context of neurological diseases like Rubinstein-Tybee syndrome, these conditions often involve abnormally low histone acetylation levels. The DNA is too tightly wound, silencing important genes.
Right, the closed state is too dominant.
Precisely. In such cases, HDAC inhibitors can be used to counterbalance this. They work by preventing the removal of acetyl groups from histones by those HDAC eraser enzymes. This promotes a more open chromatin state, which can hopefully reactivate genes that have been inappropriately silenced, offering a direct therapeutic approach. Figure 13.3 again illustrates this concept for Rubinstein-Tybee. And HDAC inhibitors are also being actively explored, as mentioned, for other conditions involving gene silencing, such as Friedreich's ataxia and Fragile X syndrome. And they're also used or being tested in certain cancers where crucial genes, like tumor suppressors, have been epigenetically silenced by deacetylation.
Okay, so inhibiting the erasers can reopen things. What about targeting methylation or the other histone marks?
Yes. A newer and very rapidly developing class of epidrugs are those that target the enzymes involved in histone methylation, the other key epigenetic mark we discussed. These are often called histone methyltransferase inhibitors, or KMT inhibitors for the writers, and histone demethylase inhibitors, or KDM inhibitors for the erasers. Now, remember we said that histone methylation marks often have more selective and precise functions compared to the broader effects of acetylation mark?
More specific signals?
Right. Because of this selectivity, inhibitors targeting these methylation writers and erasers promise to be potentially more specific in their action, and hopefully maybe less toxic systemically than some of the broad-acting HDAC inhibitors or DNA methylation inhibitors, which can have more widespread effects. And there's already been a notable success story in this area. It's an HMT inhibitor called tazemetostat. This drug has already been approved by regulatory agencies for treating specific types of rare cancers. epithelioid sarcoma, and follicular lymphoma. It works by inhibiting a specific enzyme called EZH2, which is a key component of a complex that normally adds repressive H3K27Me3 histone methylation marks, those closed signs.
So it stops the closed signs from being put up.
Exactly. Its inhibition leads to reduced levels of these repressive marks. This results in the upregulation, the reactivation of previously silenced genes. And this leads to a significant inhibition of cancer cell growth, particularly in tumors that have specific mutations that make EZH2 overactive, or where EZH2 is just generally overexpressed. It really illustrates the incredible potential for precision and effectiveness when you target specific epigenetic enzymes based on the molecular profile of the tumor. And furthermore, it's not just writers and erasers. Researchers are now actively exploring targeting readers as well.
The proteins that interpret the marks.
Yes. These are the proteins that bind to and interpret the epigenetic marks on histones and DNA, translating the code into action. For example, there are proteins with specific domains, like bromodomains, that specifically recognize and bind to acetylated histones, the open signals. By developing drugs that disrupt the ability of these reader proteins to bind to aberrant epigenetic marks, or maybe modulating their downstream activity, new therapeutic avenues are being opened. The idea here is to prevent the cell from misinterpreting a corrupted epigenetic instruction, effectively breaking the chain of command that leads from the bad mark to the disease outcome.
That's truly revolutionary, isn't it? It sounds like precision medicine, which we often discuss in terms of targeting specific genetic mutations in cancer, for example.
Yeah.
It's getting a whole new dimension through epigenetics. We're moving beyond just the static DNA sequence to targeting the complex dynamic instructions on top of the DNA.
It absolutely is. And precision oncology is probably the prime example where this shift is already transforming patient care, or at least holds that promise. The field is rapidly moving away from the older one-size-fits-all approaches of traditional cytotoxic chemotherapies. Those often come with severe systemic side effects and, frankly, might not be effective for every patient with a certain cancer type. Instead, we're transitioning towards more precise interventions, treatments tailored to the individual's specific cancer profile. And that profile now includes not just their genetic mutations, but also their unique epigenetic landscape. Large-scale omics data, things like whole genome sequencing, comprehensive epigenome maps using techniques like ChIP-seq or bisulfite sequencing, and gene expression profiles from RNA-seq. This kind of data is becoming absolutely essential. It allows for a much more precise diagnosis and enables clinicians to choose the most appropriate therapeutic strategies for that individual patient, whether it's a targeted genetic therapy, an immunotherapy, an epidrug like the ones we discussed, or perhaps a combination.
So combining genetic and epigenetic information for treatment choices.
Exactly. And this ability to stratify individuals based on their dynamic molecular profiles also makes clinical trials much more efficient. You can enroll smaller, more targeted groups of individuals who are most likely to respond to a specific epidrug, for instance, anticipating a larger effect from these personalized interventions. This represents a massive and really exciting shift in how we understand and manage disease, particularly in complex conditions like cancer. It's opening doors to truly personalized and hopefully more effective treatments down the road.
It's been an absolutely eye-opening deep dive. We've gone from the basics of the epigenome, its role in cell identity, through its dark side in diseases like cancer and neurological disorders, and now into these really hopeful new therapeutic approaches. But it also raises another big question, maybe a more personal one for all of us listening. How much control do we as individuals actually have over our epigenomes? Are we just passive recipients of our genetic and epigenetic inheritance, maybe influenced by early life? Or can our daily actions, our lifestyle choices right now, truly influence this dynamic code?
This is where that concept of the epigenome as a memory, as a dynamic record, becomes particularly powerful and I think really relevant for you, the listener. As we discussed, and the text reiterates, epigenomes vary a lot over time in response to a multitude of environmental influences. These include your diet. Your microbial encounters, the bacteria living in your gut, for example, are constantly interacting with your cells. Cellular stress, sleep patterns, physical activity, exposure to pollutants. All these factors can potentially leave their mark. It's not a fixed state. It's a highly adaptable system that's constantly recording your experiences, almost like a living diary of your health journey written in these chemical tags. And an emerging and incredibly important subdiscipline within this field is nutritional epigenetics. It falls under the broader umbrella of nutrigenomics. This field actively explores how specific dietary compounds, the nutrients and non-nutrients in your food, directly affect your epigenome. And the molecular link is actually quite direct, quite tangible. Many of those writer and eraser enzymes for epigenetic marks, they use intermediary metabolites as essential co-substrates or cofactors. They need them to function.
Metabolites,
Yeah.
like from breaking down food.
Exactly. These metabolites are often derived directly from the food you eat. For instance, a crucial molecule called acetyl-CoA. It's produced when your body breaks down carbohydrates and fats. Well, acetyl-CoA is the key donor molecule needed by the HAT enzymes that add acetyl groups to histones, the ones that open up genes.
So more carbs and fats might mean more potential for acetylation.
It's more complex than a direct one-to-one, but yes, substrate availability matters. Similarly, other metabolites derived from amino acids, like S-adenosylmethionine or SAM, are essential for DNA and histone methylation. Vitamins like folate are crucial too. This means that what you eat can literally and directly influence the activity of these epigenetic writers and erasers. It can subtly, or sometimes maybe profoundly, reshape your epigenome over time. And research is actively studying specific compounds found abundantly in healthy foods, fruits, vegetables, teas, spices, things like genistein from soy, resveratrol from grapes, curcumin from turmeric, various polyphenols from green tea. They're being studied for their remarkable ability to modulate these epigenetic enzymes and transcription factors, with potential implications for disease prevention and maybe even therapy.
So you are what you eat gets a whole new molecular meaning here.
It really does. And our bodies have also evolved sophisticated nutrient-sensing pathways that integrate these nutritional signals and, in turn, influence the epigenome. Pathways like the insulin-IGF signaling pathway, which senses glucose levels, or the 2R pathway, which senses amino acid availability. These are prime examples. They act like central hubs, connecting your nutritional and energetic status directly to cellular processes, including the gene expression patterns controlled by the epigenome. For example, calorie restriction, simply reducing food intake without causing malnutrition, has been consistently shown to extend lifespan in various model organisms, from yeast and worms up to mice.
We hear a lot about calorie restriction.
Right, and what's truly interesting is that the underlying regulatory principles involving certain enzymes like SIRTs and AMPK, which sense energy levels and influence the epigenome, these seem to be surprisingly similar in humans. This offers a powerful potential avenue through dietary modulation for promoting healthy aging and preventing age-related diseases like type 2 diabetes, cardiovascular diseases, and maybe even cancer.
Okay, so diet is huge. What about exercise? Does moving our bodies also rewrite our epigenome?
Absolutely. Physical activity's impact on your epigenome is also significant and increasingly well documented. Regular exercise, for instance, can induce profound changes in the chromatin structure, the epigenetic landscape of your skeletal muscles. Studies have clearly shown it can increase levels of histone acetylation in muscle genes, making them more active and responsive. It can also influence the cellular localization and activity of certain HDAC eraser enzymes. This provides clear molecular evidence for how lifestyle choices, like incorporating regular physical activity, are actively writing your epigenetic story every day. It contributes to healthy aging, improves metabolic health, and potentially prevents the onset of many age-related diseases. This includes metabolic disorders like type 2 diabetes, cardiovascular diseases, and there's even evidence suggesting it reduces risk for certain cancers, partly through these epigenetic mechanisms. It's truly empowering, I think, to know that your daily choices have this profound, direct molecular effect on how your genes function.
It really is. So it's definitely not just about the genes you inherit from your parents, that fixed blueprint we started with. It's critically about how your daily choices, your environment, maybe even things that happened before you were born or to previous generations, how all that interacts with those genes through this incredibly dynamic and responsive epigenetic layer. That's such a profound shift in how we should think about personal health and disease susceptibility. Makes that old debate of nature versus nurture seem almost obsolete, doesn't it? It's really nature through nurture, mediated by the epigenome.
Exactly. That's a great way to put it. And the source material highlights this clearly, stating that epigenome-wide changes, both those occurring prenatally during development and those accumulating throughout adult life, play a significant role in the development of common complex diseases like type 2 diabetes. And the rapid development of these powerful omics technologies we mentioned, leading to what are called integrated personal omics profiles, or IPOP.
IPOP, so combining genomics, epigenomics, transcriptomics.
Exactly. Maybe proteomics, metabolomics too. getting a dynamic snapshot of an individual's biology over time, not just their fixed genes. This is already allowing us to see these dynamic individual changes in response to life events or interventions, sometimes in real time. For instance, some pioneering IPOP studies have successfully detected the very early molecular signs of type 2 diabetes outbreaks in individuals before clinical symptoms might even appear. And remarkably, they've also shown how these early disease trajectories can sometimes be controlled or even reversed through highly personalized diet and exercise interventions tracked at the molecular level.
Wow, so you can see the intervention working on the omics level.
Potentially, yes. This ability to stratify individuals based on their dynamic molecular profiles, their specific genetic background, coupled with their ever-changing epigenome, their gene expression patterns, their metabolic state. It means we can realistically move towards truly personalized nutrition and highly targeted preventative strategies and therapies. This approach allows us to use diet not just for general well-being, but as a precision tool to preserve health. And, when necessary, combine it smartly with synthetic drugs, perhaps including epidrugs, for more effective and individualized treatment based on a person's complete molecular picture. This fundamentally changes everything about how we understand and manage health and disease prevention. It moves us closer to a future where medicine is truly tailored exactly to your unique dynamic biology.
So what does this all mean for us? Where do we stand after this deep dive? Today, we've explored the epigenome, that incredibly dynamic, really adaptable layer sitting right on top of our DNA. It profoundly shapes how our genes are expressed and ultimately how our bodies function, develop, and age. We've seen its fundamental role in establishing and maintaining cell identity, making sure our diverse cells do their proper jobs. We've explored its profound and often driving impact when it gets reprogrammed incorrectly in devastating diseases like cancer. We saw how epigenetic errors can silence vital tumor suppressors or activate growth programs. And we learned about its critical influence on neurological disorders, affecting everything from brain development to potentially storing our long-term memories. And maybe most excitingly, we've touched upon the revolutionary promise of new therapies, epidrugs, that aim to actually correct these epigenetic errors, opening up a whole new frontier in precision medicine, moving beyond just reading the fixed genetic code.
Yeah, and if we connect this to the bigger picture, the epigenome really represents this powerful, potentially modifiable dimension of our biology. It goes far beyond the fixed sequence of our DNA that we inherit. It provides a crucial explanation for why we're so much more than just our inherited genes. It helps explain individuality, even between identical twins, and it offers completely new avenues for understanding individual disease susceptibility and, perhaps more importantly, for developing highly personalized interventions, preventative or therapeutic. It's a profound reminder, I think, that biology is rarely static. Our bodies are constantly responding, adapting, and recording our interactions with the world around us, right down to the molecular level.
So, as you go about your day-to-day, maybe just consider how your everyday choices, from what you choose to eat for lunch, whether you take the stairs or the elevator, the environment you live and work in, even how you manage stress. Consider how all these things might be actively writing and rewriting the story of your epigenome. It's a story that influences not just your own health trajectory, your susceptibility to conditions like cancer and neurological diseases, but potentially through mechanisms we're only beginning to understand, maybe even the health of future generations. What incredible new insights will we uncover as we continue to map these complex and dynamic landscapes? And more importantly, how will we harness this knowledge to truly reshape human health and longevity in the future? This has been the deep dive into epigenetics and disease for the Genomic Medicine program. Thank you for joining us on this journey to becoming more well-informed.